Hey everyone, and welcome back for another deep dive. Today we're going exploring these fascinating molecules called hemoproteins.
Specifically hemoglobin and myoglobin.
Hemoglobin and myoglobin. You know them, right? The
Hmm.
oxygen carriers in our bodies.
Yeah, exactly.
But get this, listeners.
There's way more to these molecules than just that.
So much more. And our source material seems to be like straight out of some biochemistry notebook.
Yeah, it really dives deep into the nitty-gritty.
All these crazy details about these proteins.
It's fascinating stuff. And one thing that really jumps out is that both hemoglobin and myoglobin rely on this special molecule called heme to do their thing.
Bean.
Heme, think of it like a molecular stage where iron is the star performer.
Okay, I'm picturing it. So iron's like center stage.
Right at the center.
And the notes describe it as this complex structure with iron right at the center.
Held in place by...
Held in place by 4 nitrogen atoms. It sounds like iron is really the key player here.
Absolutely.
Yeah.
But here's the thing. That same iron-centered structure can do totally different things depending on the protein environment it's in.
Really?
Yeah.
So it's not just about the heme itself,
What's the whole context?
but also its surroundings. That's fascinating.
Exactly. For instance, in cytochromes, heme acts like a tiny shuttle moving electrons around.
Okay.
But in an enzyme like catalase, it helps break down hydrogen peroxide,
Which is...
which is harmful to our cells. Yeah.
Gotcha. So it can play different roles.
Right.
Different functions.
Hemoglobin and myoglobin, they use heme to bind oxygen.
Okay.
And in a way that's reversible, which is something you don't see often with iron in biological systems.
Interesting. So it's like heme is a multi-talented actor playing different roles depending on the script.
You got it.
I like that analogy. But in our case, it's all about oxygen.
All about oxygen.
The notes talk about myoglobin as the oxygen hoarder in our muscles.
Oh, yeah. That's the vibe.
Stores oxygen and helps it move around inside muscle cells. So it's like a dedicated oxygen supply just for our muscles.
Yeah, like a little reservoir.
Right.
Exactly.
And what's cool is that myoglobin is structurally similar to a single unit of hemoglobin, almost like a simplified model.
Yeah, a great way to think about it. Understanding myoglobin helps us understand the more complex hemoglobin. Myoglobin's a compact molecule. About 80% of its structure is made up of these neatly coiled sections called alpha helices.
Alpha helices.
Kind of like a tightly wound rope.
Okay, I'm picturing a rope.
Right.
And the notes mention something about proline and other elements creating this compact structure. Are those like the knots in our rope analogy?
Ooh, I like that. You could think of it that way. These elements interrupt the smooth coiling of the alpha helices,
So they're causing...
creating bends and loops that help the protein fold into its final compact shape.
Gotcha. So it's not just a straight rope.
No, it's all bundled up.
It's got some
Yeah.
bends and twists to it.
Another clever design feature is the arrangement of amino acids. The ones that don't like water, the hydrophobic ones, are all huddled together on the inside, providing stability.
Those are like tucked away.
Avoiding the water at all costs.
Exactly. And then the charged amino acids, on the other hand, are on the outside interacting with water.
Interesting.
So it's a well-organized structure with everything in its place.
I like it organized, but where does heme fit into all of this?
Heme sits snugly within the myoglobin molecule
Okay,
in a kind of hydrophobic pocket.
a pocket.
And 2 histidine residues are crucial here. One binds directly to the iron in heme, anchoring it in place.
Okay.
The other helps stabilize oxygen binding, making sure that oxygen can bind and release without oxidizing the iron.
Wait, so the iron in the heme doesn't rust?
Yeah, that's a great question.
Because isn't that what usually happens to iron when it's exposed to oxygen? It rusts?
You'd think it would, wouldn't you? But the protein environment surrounding the heme actually prevents that.
Really?
It creates a special microenvironment where oxygen can bind to the iron reversibly without causing it to fully oxidize into rust. Wow, that's pretty amazing. It's like the protein is shielding the iron from rusting.
Exactly. It's a delicate balance that allows for this reversible oxygen binding, which is essential for myoglobin's function.
It's a pretty impressive system.
It is.
So we've got myoglobin, the oxygen hoarder, in our muscles.
Right.
Now let's talk about hemoglobin, the oxygen shuttle.
The transporter.
Found in our red blood cells, its job is to transport oxygen from our lungs to the rest of our body. And unlike myoglobin, which works solo, adult hemoglobin is a team player.
Yeah, it's a team effort.
It's made up of 4 chains.
4 polypeptide chains.
2 alpha and 2 beta working together.
That's right. We call this a tetramer.
A tetramer.
4 parts working as one.
Okay.
And this teamwork is really what makes hemoglobin so remarkable.
The notes mention a quaternary structure.
Ah,
What
yes.
exactly does that mean?
Think of it as how these 4 chains are arranged and interact with each other.
Okay.
In hemoglobin, it's like 2 sets of identical twins. The alpha-beta pairs linked tightly together. They're held together by strong bonds, like 2 hands clasped together,
Ah.
and weaker bonds between the pairs, allowing for flexibility.
Okay, so there's a balance between strong bonds within the pairs and weaker bonds between them, which allows for movement. Why is this flexibility important for oxygen transport?
It's all about efficiency.
Yeah.
Hemoglobin needs to be able to bind oxygen tightly in the lungs, where there's plenty of it,
Yeah.
and then release it easily in the tissues where oxygen is needed.
Right, where it's needed.
And this flexibility allows it to do just that. Hemoglobin actually exists in 2 forms,
2 forms,
a taut or T form and a relaxed or R form.
T and R.
When oxygen binds to hemoglobin, it triggers a shift from the T form to the R form,
Okay.
increasing its affinity for oxygen.
So the more oxygen it carries, the more it wants to grab onto.
Exactly.
That's incredible.
It's a very elegant system.
But how does our body know how much oxygen to pick up and deliver?
That's where things get even more interesting.
Yeah.
Remember those oxygen dissociation curves we talked about?
They...
They show how well a protein binds to oxygen at different oxygen levels.
Okay.
Myoglobin, which only has one binding site for oxygen, has a simple hyperbolic curve.
Right.
But hemoglobin, with its 4 chains and 4 binding sites, has a more complex sigmoidal curve.
Oh, wow.
This difference is due to something called cooperative binding.
Cooperative binding. Tell me more about that.
All right, imagine it like this. When one oxygen molecule binds to hemoglobin,
Yeah.
it makes it easier for the next one to bind and so on.
Oh.
It's like a chain reaction.
Like a domino effect.
Exactly. This cooperative binding allows hemoglobin to load up on oxygen in the lungs, where oxygen is abundant,
Okay.
and then efficiently unload it in the tissues, where oxygen levels are lower.
So it's like hemoglobin has a built-in sensor for oxygen levels, releasing just the right amount where it's needed most.
You got it.
This cooperative binding is crucial for efficient oxygen delivery. Could myoglobin, with its single binding site, achieve this same level of precision?
No, not really, because myoglobin's structure and single binding site mean that it holds onto oxygen very tightly, regardless of the surrounding oxygen levels.
Oh.
It wouldn't be able to fine-tune oxygen delivery the way hemoglobin can.
Okay.
This is where the beauty of hemoglobin's complexity really shines.
I see that.
And it doesn't stop there.
Yeah.
To make this delivery system even more sophisticated, we have allosteric effectors.
Allosteric effectors.
These are molecules that can fine-tune hemoglobin's behavior
Okay.
by binding to it at sites other than the oxygen binding sites.
So they're like modifiers.
Yeah, think of them like dimmer switches for oxygen binding.
Interesting. So it's not just about oxygen itself, but also about these other factors that can influence how hemoglobin behaves.
Exactly.
The notes mention pH, carbon dioxide, and a molecule called 2.3 BPG as the key players here.
Yes, those are the big ones.
Let's start with pH. How does that affect things?
Well, when our tissues are working hard,
Yeah.
they produce more carbon dioxide and lactic acid, which increases acidity. In other words, the pH goes down. This lower pH, along with the higher carbon dioxide levels, actually makes hemoglobin less sticky to oxygen.
So less likely to hold on to it.
Exactly.
Interesting.
This is known as the Bohr effect.
De Boer effect.
So when our muscles are working hard and screaming for more oxygen,
They need more.
the Bohr effect steps in and tells hemoglobin to let go of oxygen more easily.
Wow, that's a pretty cool system. What's the evolutionary advantage of having this mechanism? Why not just have hemoglobin release oxygen at a constant rate?
Well, if hemoglobin released oxygen at a constant rate, it wouldn't be able to respond to the changing needs of our body.
Ah, I see.
The Bohr effect allows for a dynamic response, ensuring that oxygen is delivered where it's needed most when it's needed most.
Okay, so like when I'm exercising,
That's all
my
about-
muscles need more oxygen
Than when you're just sitting around.
than when I'm just chilling on the couch.
The Bohr effect helps meet that increased demand.
That makes so much sense. It's like our bodies have this incredible system of checks and balances to make sure everything runs smoothly. What about that 2003 BPG molecule?
Yeah.
What role does it play in all of this?
2003 BPG is another fascinating player in this story.
Okay.
It's produced during glucose metabolism
Uh-huh.
and binds to hemoglobin, reducing its affinity for oxygen.
So it's making it even less sticky.
Exactly, and this is especially important in situations like high altitude or anemia, where oxygen availability is limited.
So 2003 BPG is like a backup system, ensuring oxygen delivery even when things get tough.
You could say that.
What I find fascinating is that our bodies can adapt to these challenging environments. Like,
Yeah.
how does that even work?
Well, think about it this way. When you go to a high altitude where the air is thinner,
Yeah.
your body senses the lower oxygen levels
Okay.
and responds by increasing 2003 BPG production.
So more 2003 BPG.
This helps hemoglobin release oxygen more readily in the tissues, compensating for the reduced oxygen availability.
So it's like a
It's a brilliant adaptation
So it's a brilliant compensation mechanism.
that allows us to function even in challenging conditions.
That's incredible. It's like our bodies are constantly monitoring and adjusting to make sure we get the oxygen we need. So far, we've been talking about normal adult hemoglobin.
Right.
But the notes mention that not all hemoglobins are created equal.
Ah, yes.
There's also fetal hemoglobin, or HbF,
HbF.
which has a higher affinity for oxygen compared to adult hemoglobin, or HbA.
Right.
What's the reason for this difference?
Well, a fetus relies on its mother's blood supply for oxygen.
Of course.
Fetal hemoglobin's higher affinity for oxygen allows it to effectively steal oxygen from the mother's hemoglobin.
So it can outcompete it?
Exactly, ensuring the developing fetus gets the oxygen it needs.
That is so clever.
Pretty neat, huh?
It's like the fetus has its own specialized oxygen-grabbing tool.
You got it.
What happens after the baby is born? Does it keep using fetal hemoglobin?
Well, after birth, when the baby starts breathing on its own,
Right.
the need for this super strong oxygen grab isn't as important.
Makes
So the body gradually switches from producing fetal hemoglobin to producing primarily adult hemoglobin.
So it's like we're
It's another example of our bodies adapting to different stages of life.
switching over to the adult version?
Exactly.
The notes also mention other minor hemoglobins like HbA2 and HbA1c. What can you tell us about those?
HbA2 is a normal variant of hemoglobin that makes up about 2–3% of the hemoglobin in adults.
Okay.
It's not as well understood as HbA, but it seems to play a minor role in oxygen transport.
Okay.
HbA1c, on the other hand, is a form of hemoglobin that's linked to blood sugar levels.
Oh, interesting.
It's formed when glucose molecules attach to hemoglobin.
So HbA1c is like a marker of blood sugar levels.
Exactly. The amount of HbA1c in your blood reflects your average blood sugar levels over the past 2 to 3 months.
Oh, wow. So it's like a long-term indicator.
Exactly. This is why HbA1c is a valuable tool for monitoring long-term blood sugar control in people with diabetes. It gives doctors a much better picture of how well their patients are managing their blood sugar than a single blood glucose reading would.
That's a really clever way to track blood sugar levels over time. But speaking of things going wrong, the notes also mention these things called hemoglobinopathies.
Hemoglobinopathies.
They're genetic disorders caused by problems with hemoglobin production.
Right.
The most well-known one is sickle cell anemia, right?
Yes, sickle cell anemia is a classic example of a hemoglobinopathy. It's caused by a single mutation in the gene that codes for the beta-globin chain of hemoglobin.
And this one tiny change can have such huge consequences.
Oh, absolutely.
Tell me more about it.
So in sickle cell anemia, a single amino acid, glutamic acid, is replaced by valine in the beta-globin chain.
Okay.
This seemingly minor substitution creates what we call a sticky patch on the hemoglobin molecule.
A sticky patch, what does that mean?
It means that this part of the molecule doesn't like to be around water.
Oh.
And this causes the hemoglobin molecules to clump together, especially when they release oxygen.
Okay.
These clumps then form long, rigid fibers inside the red blood cells, distorting them into a sickle shape.
So this one tiny change in the amino acid sequence triggers this whole cascade of events,
That's a domino effect.
leading to these misshapen red blood cells.
Yeah.
What are the consequences of having these sickle-shaped cells?
So these sickled cells are less flexible
Okay.
and can get stuck in small blood vessels blocking blood flow.
Oh, no.
This can cause a whole host of problems, including pain tissue damage and even organ damage.
Wow.
It's a serious condition that highlights how even the smallest changes in protein structure can have profound effects on our health.
It's both fascinating and terrifying at the same time. So you've got these sickle-shaped red blood cells causing all sorts of problems. Are there other types of hemoglobinopathies besides sickle cell anemia?
Yes, there are other hemoglobinopathies, each with its own unique set of challenges.
Like what?
For instance, there's hemoglobin C disease, which is caused by a different mutation in the beta-globin gene.
Okay.
And then there are the thalassemia syndromes, which involve reduced or absent production of one or more of the globin
Oh,
chains.
there's a whole range of these disorders.
There are.
This is all so much to take in.
It is a lot.
It's incredible how these seemingly simple molecules, these hemoglobins, are so central to our existence and how even the smallest changes can have such a huge impact on our health.
It really is remarkable, and we've only just scratched the surface.
There's so much more to learn.
So much more.
Well, I'm definitely ready to dive deeper, but before we do, let's take a moment to reflect on what we've learned so far. What stood out to you, listeners? What sparked your curiosity? Perhaps it's the intricate structure of these molecules or the way they adapt to different environments, or maybe it's the devastating consequences of even the smallest genetic errors. Whatever it is, hold on to that thought as we continue our exploration of hemoglobin and myoglobin.
And we'll be back soon with part 2.
And stay tuned.
Welcome back to our deep dive into the world of hemoglobin.
It's wild to think about how those tiny changes at the molecular level can have such huge effects on our health.
It really is, and it shows how everything in our bodies is connected.
Totally. Speaking of connections, the notes go into how those globin genes are organized. They're like blueprints for those globin chains that form hemoglobin.
Right, like the instructions.
So we've got these globin genes, but how do they actually become those globin chains we need?
It's a pretty amazing process. It starts in the nucleus of red blood cell precursors,
Red blood cell precursors.
the cells that eventually become red blood cells.
Got it.
The first step is called transcription, where the globin gene's instructions are copied into a molecule called messenger RNA or mRNA.
Okay, so if the globin gene is like the master recipe, the mRNA is like a copy someone makes to actually use in the kitchen.
That's a great analogy.
Mhm.
And just like you might tweak a recipe a bit before using it,
Right. Add a little spice.
the initial mRNA copy goes through some editing.
Editing.
It contains extra bits called introns that need to be removed.
Introns.
This editing process is called splicing.
Mm.
Think of it like removing the bloopers from a film reel to get the final cut.
So it's like refining the recipe before using it to bake the cake.
Exactly. Once those introns are removed and the remaining pieces called exons are joined together, we have our mature mRNA ready for action,
Mature mRNA.
ready to go.
So what happens next?
This mature mRNA molecule then leaves the nucleus
Okay.
and heads into the cytoplasm, the busy part of the cell where proteins are made.
Okay.
Here it encounters ribosomes, the protein-making machinery of the cell.
Ribosomes.
The ribosomes read the mRNA's instructions.
And what the chefs?
Yeah, and assemble a chain of amino acids according to the recipe, creating a brand new globin chain.
It's amazing how this whole process from gene to protein is so intricate and yet so precise.
It's a beautiful dance of molecular machinery.
For sure. But let's get back to those hemoglobinopathies. We talked about sickle cell anemia, but the notes also mention thalassemia syndromes. What can you tell us about those?
Thalassemias are a group of inherited blood disorders where the problem isn't abnormal hemoglobin like in sickle cell anemia.
Okay.
Instead, there's just not enough normal hemoglobin being produced.
So instead of a misshapen protein, it's more of a quantity issue.
Precisely.
Is that because of a problem with those globin genes we were just talking about?
Exactly. And the severity of thalassemia depends on which globin chain is affected and to what extent.
Okay.
The notes specifically mention beta thalassemias, which affect the production of beta globin chains.
Okay, so to recap, normal adult hemoglobin has 2 alpha globin chains and 2 beta globin chains.
Right, 2 of each.
So what happens if there aren't enough beta chains?
If there aren't enough beta chains, the excess alpha chains can't form stable hemoglobin molecules.
Huh.
They end up clumping together, causing damage to the red blood cells.
Not good.
It's like trying to build a house with too many bricks and not enough wood. The structure becomes unstable.
That's a good analogy.
And this leads to problems for those red blood cells.
What kind of problems?
They become fragile and break down prematurely, leading to anemia.
Oh, so that's why people with thalassemia often have anemia.
Exactly.
The notes mention that the severity of beta thalassemia depends on whether one or both copies of the beta globin gene are defective. Why is that?
Remember, we inherit 2 copies of each gene, one from our mother and one from our father.
Right.
In beta thalassemia, if only one copy of the beta globin gene is defective, call it beta thalassemia minor.
Okay.
People with this condition often have mild anemia or sometimes no symptoms at all.
So it's like having a backup copy of the gene that can partially compensate for the defective one.
Exactly.
But what if both copies are defective?
That's when things get more serious. It's called beta thalassemia major. also known as Cooley's anemia,
Cooley's anemia.
because there's no functional beta-globin gene. Very little normal hemoglobin can be made.
Oh, wow.
This is a much more severe condition that often requires regular blood transfusions to provide the body with enough healthy red blood cells,
So in that case, they're basically relying on external sources of hemoglobin.
essentially.
That makes sense. It's all about how many functional copies of the gene you have.
What about alpha thalassemias? Do they work the same way? The principle is similar, but with alpha thalassemias, it's the production of alpha-globin chains that's affected. And here's where it gets a little more complex. We actually have 4 copies of the alpha-globin gene,
4.
2 on each chromosome 16.
Wow.
This means there's a wider range of severity depending on how many of those genes are defective.
So you could have 1, 2, 3, or even all 4 copies of the alpha-globin gene affected.
That's right.
That seems like it would create a lot of variability in how the disease presents.
It does. If only 1 or 2 alpha-globin genes are defective, the effects are usually mild.
Okay.
However, if 3 genes are defective, it results in a condition called hemoglobin H disease, which causes moderately severe anemia.
And if all 4 are defective?
If all 4 alpha-globin genes are defective or missing, it leads to a condition called hemoglobin Bart's hydrops fetalis.
Hemoglobin Bart's hydrops fetalis. That sounds very serious. What are the consequences of having no functional alpha-globin genes?
It is very serious.
Yeah.
Remember how fetal hemoglobin needs alpha-globin chains? Without them, the fetus can't make enough fetal hemoglobin, and that leads to severe anemia and other complications. Sadly, hemoglobin Bart's hydrops fetalis is often fatal either before or shortly after birth.
That's really sad. It's remarkable how these different types of thalassemia, depending on the specific genetic defects, can lead to such a wide spectrum of severity. It really underscores the importance of genetic testing and counseling for families with a history of these disorders.
I completely agree. Early diagnosis and management can make a huge difference in the lives of people affected by thalassemia.
Absolutely. Okay, so we've covered sickle cell anemia and thalassemia syndromes, both of which involve problems with the globin chains themselves. Are there other ways things can go wrong with hemoglobin?
There are sometimes the problem isn't with the globin chains, but with the heme part of the molecule. The notes mention a condition called methemoglobinemia. Remember how we talked about the iron and heme needing to be in the correct state, the ferrous state, to bind oxygen?
Right, and you said that the protein environment around the heme prevents the iron from rusting.
Exactly. Well, in methemoglobinemia, the iron in the heme is oxidized to the ferric state, and it can't bind oxygen.
So it's like the iron in the hemoglobin actually does rust even though it's inside the protein?
You could say that.
But I thought the protein was supposed to prevent that.
It usually does, but sometimes things go wrong.
Ah,
This oxidation can be caused by certain drugs' exposure to specific chemicals or even genetic defects that affect the structure of the hemoglobin molecule itself.
so there are both acquired and inherited forms of methemoglobinemia.
That's right.
What kind of symptoms does it cause? Do people literally turn rusty colored?
Not quite rusty colored, but they do develop a bluish discoloration of the skin and mucous membranes called cyanosis.
Cyanosis.
This happens because methemoglobin has a different color than normal hemoglobin.
So people with methemoglobinemia literally turn blue.
They can?
That must be pretty startling.
It can be, and the severity of symptoms depends on how much methemoglobin is in the blood. Mild cases might only cause a slight bluish tinge, while severe cases can lead to shortness of breath fatigue, headache, dizziness, and even seizures or coma.
So it can range from a cosmetic issue to a life-threatening condition. What kind of treatment is available?
The treatment depends on the cause and severity. For mild cases, simply avoiding the offending drug or chemical might be enough.
Okay.
But for more severe cases, a medication called methylene blue is often used.
Methylene blue? How does that work?
Methylene blue acts as an electron donor, which means it can provide electrons to convert the ferric iron in methemoglobin back to the ferrous iron, restoring the hemoglobin's ability to carry oxygen.
So it's like a little helper molecule that comes in and fixes the rusty iron.
Exactly.
That's pretty cool, and it just goes to show how intricate the chemistry of hemoglobin is.
It really does.
This deep dive has certainly been a whirlwind of information. We've covered so much ground, from the structure and function of hemoglobin and myoglobin to the mechanisms of oxygen binding and transport, the fascinating role of allosteric effectors, and the complexities of hemoglobinopathies. It's amazing how much is happening at the molecular level constantly and flawlessly to keep us alive and functioning.
It is truly remarkable, and we've only just scratched the surface. There's a whole universe of knowledge waiting to be discovered within these seemingly simple molecules.
Well, on that note, let's pause here for a moment to let all this information sink in. When we come back for the final part of our deep dive, we'll wrap things up with some final thoughts and perhaps some mind-blowing takeaways that will leave you pondering the wonders of these remarkable molecules. What are your initial thoughts, listeners? What aspects of hemoglobin and myoglobin intrigue you the most? And we're back for the final part of our deep dive into hemoglobin and myoglobin.
It's crazy to think we walk around with these tiny molecules doing all this amazing stuff inside us.
Yeah, they really are the unsung heroes of our bodies. The notes make a big deal about how perfectly hemoglobin is set up to deliver oxygen exactly where it's needed.
Right, it's like everything about it is fine-tuned for that job.
The cooperative binding, the effects of pH and CO2, even how it changes during different stages of life, it's like a well-oiled machine.
It really is. And don't forget, it also helps transport carbon dioxide back to the lungs to be exhaled.
Oh, right, so it's keeping our pH balanced too.
Exactly, a true multitasker.
Like the ultimate delivery service, bringing in the good stuff and taking out the trash. But like we've learned, things can go wrong.
Unfortunately, yeah.
Like with those genetic disorders, sickle cell anemia and thalassemia.
Makes you realize how fragile these systems can be.
Definitely. And it shows how important it is to understand them at the molecular level so we can develop better treatments.
Absolutely. There's so much potential there for future research.
Like what kind of stuff?
Well, for example, scientists are looking at ways to reactivate fetal hemoglobin production in people with sickle cell disease.
Oh, that's interesting.
The idea is if you could boost fetal hemoglobin, which doesn't sickle, you could lessen the severity of the disease.
That makes sense.
And in gene therapy, researchers are working on correcting the genetic defects that cause thalassemia.
So there's hope for actual cures.
It's an exciting time for sure. There's real progress being made.
That's awesome to hear. This whole deep dive has given me a new respect for hemoglobin and myoglobin and how crucial they are for life. What about you, listeners? What are you taking away from all this?
Maybe it's made you more curious about the molecular world.
Or inspired you to learn more about genetics?
Perhaps it's even sparked a new appreciation for the amazing processes happening inside your own body right now.
That's a great point. Well, we've come to the end of our deep dive, but the exploration doesn't have to stop here.
There's always more to learn and discover.
So keep those minds curious, everyone.
And keep asking questions.
That's what the deep dive is all about. Until next time.